Okay. All right. So action items from the previous meeting. We decided to get rat PK for M4K 2009. That's done. I'll show you the results. We wanted to test M4K9 in vivo, looking at IP dosing. This came up as a part of a discussion based on Jerome's in vivo model that it might be more convenient to dose the compound IP rather than oral because the mice are pretty young in his model. So we've done that, and I'll show you the results. They look pretty good. KCO2 data for M4K 3007, that was done. Sue, you have those results. We do see that the compound effluxes a little bit, which might explain why we don't have good brain penetration with that compound, but the PK is still pretty good. So we wanted to follow up with full IC50s for the relevant kinases that we identified in the kinome screen for M4K 2009, and also follow up on the serum findings. We've done that. I'll show you the results in my part of the presentation. We ran an MTD study, a maximum tolerated dose study here at OSER, and we were going to analyze the plasma and brain samples and finish that study. That's still ongoing. We have some preliminary results that look quite promising. Brain penetration looks really good. We're just looking at the data a little bit more carefully before we release it, looking at absolute concentrations just to make sure that we're all aligned. We wanted to select 5 additional compounds for mouse PK experiments, and David and I have selected those and sent them out. We don't have results, but they should be dosed next week. We decided to get brain penetration data for 2117, 2118, and we added another compound, 2143. I'll show you those compounds and their profile in my part of the presentation. We're always updating the Xray list, and I think we'll have a couple of additional compounds to add this month. The MTA with Angle and related to the DIPG cell lines and models, I think this is still something that's ongoing. The MDCK MDR1 is still not up and running yet, but I think we're still working on trying to get that up and running. But again, as we mentioned, the KCO2 permeability does seem to be pretty predictive, at least for this series of molecules that we're working in. So I think that's a good sign. And then we talked about trying to extend the anti-proliferation assays to longer time points based on the SH data that we saw for M4K or for ALK2 and knocking down ALK2. It takes about 11 days to get all the way to bring the curve all the way down using SH, and so it would be good to actually look at proliferation in a longer time point. And I'll revisit that and the reasons why during my part of the presentation as well. I did just want to reiterate that we do have the M4K database and compound progression tool up and running now. We're able to provide SD files or Excel files to anyone who wants them, who's interested in looking at all of the data that we've been able to generate to date in the project for all of the compounds. The only data that's not in there are the external data that we've generated based on hERG, the pharmacokinetics that we ran at both Farmeron and Charles River. That data is not in the database yet, or basically the hERG. So we're working on that. We'll update the database when we can, but it's still a relatively limited number of compounds, so I think it's still quite easy to follow. And I think we'll talk about this later on, but we want to get some version of one of these files uploaded to the M4K website on a routine basis so that people can visit the site and download the file and be able to follow and track the SAR themselves. Okay, so with that, I'm just going to start right into the project summary. So I can't believe it's been a year already. Time goes very quickly, and I think we've made some great progress over the first year, and hopefully we can match that progress in the second year. But certainly, and those of you who have been attending all of the meetings know I like to show this slide. I think it was Alex who generated it, but DIPG, there's definitely a dire unmet medical need. Very, very low 5-year survival rate. Essentially, no one survives. And so, I think what we're doing is very important. I think it's, I think it has the potential to make a big difference in these patients' lives. And, I think that, time is of the essence, and let's keep pushing this project as far as possible to get to the answer. So we all know then that the target we're going after is ACVR1, ALK2. It's a mutation that causes tumors in the pons region of the brain. And ACVR1 was first shown to have activating mutations in DIPG several years ago now, and it's been since demonstrated that about 25 to 30% of all DIPGs have these ACVR1 mutations. SH knockdown of ALK2 appears to decrease the proliferation of DIPG cell lines. It does take this 11 days to bring it all the way down, though, and that's, again, something that we need to keep in mind. ALK2 mutation coupled with histone mutations and some others that Jerome has talked to us about can lead to DIPG-like tumors in mice. And the starting point for this project, this LDN-214117, this molecule was an orally bioavailable, brain-penetrant compound that demonstrated modest efficacy in a DIPG in vivo model. And so this is kind of the high-level overview of why we think going after this target can lead to an improved treatment for these DIPG patients. I won't go over the biology very much other than to say that inhibiting ALK2 should result in decreased levels of phospho SMAD 1, 5, and 8 in the cells, and that should lead downstream into decreased expression of the target genes of this, which is ID1 and ID2, among several others, many others actually. But these are two that we think are important. And I know, Jerome, you have a nice
there's a lot of them, but definitely, ID1 and ID2 are kind of like the canonical target.
Exactly, right? And I think Jerome actually has a much nicer figure than this that eventually we'll be able to show once it gets published. With that, so the key issues to address that we thought of when we first started this project for a drug discovery campaign. So the freedom to operate, there were 2 patents that covered this LDN-214117. They were from Harvard. And what we wanted to do was to try to build molecules that were outside of the scope of those patents so that we would have freedom to operate. Now, given the limited market potential of this target, we don't think that it's that big a deal to actually have to have freedom to operate, but in order to simplify our lives, we all think it's a better idea to move forward with a molecule that isn't covered by someone else's patents. Less paperwork, exactly, yeah. But you'll see when I start showing you the data that, LDN-214117 is selective, really quite selective for ALK2. It does have this good pharmacokinetics, good brain penetration, but it really isn't as potent as we think we need to be. And then, we do want to maintain or improve the selectivity versus ALK5, certainly. This has been linked to cardiotoxicity in the clinic and in several other projects. And so, there's some evidence coming out in the literature that this may not be the case, but, at least initially, this is what we're targeting, the selectivity against these other ALK kinases, specifically ALK5 and ALK4. And because it's a brain target, the compounds do need to cross the blood-brain barrier. The lead molecule, 214117, does cross the blood-brain barrier. There's data out there suggesting that it crosses quite well. And we were planning on looking at CNS and multi-parameter optimization scores, and kind of optimizing the molecular weight, polar surface area, limiting the number of hydrogen bond donors and acceptors, keeping the log P within a good range, and and the log D to try to get the best chance possible to have our compounds have good exposure in the brain. And then, as I mentioned earlier, certainly time is of the essence. There's this dire unmet medical need. There are other targets that we could go after in this disease. EZH2 is one that has some link to the disease. PDGFR-alpha is also another possibility where there's been some links made. So, you know, the team really decided to focus on generating an early proof of concept, try to demonstrate that an ALK2 inhibitor can impact this disease as soon as possible. And so I think we've taken a lot of steps in that direction. And I think you'll see that through this presentation. So we wanted to initiate chemistry, and you know, the lead molecule was actually quite good. It had a lot of very good properties about it, and so we decided that, you know, we would try to stay close to the lead structure, but just make subtle changes to try to improve the potency, but maintain the selectivity and the good PK and good brain penetration properties that are inherent with that lead molecule. We did think that replacing this trimethylphenyl, because it could be a potential metabolic liability, you can dealkylate these. It's a very electron-rich ring, which is never that great. But, you know, in this particular case, it has proven to be quite challenging. And the molecules that we're generating appear to be well tolerated. They have good pharmacokinetics. And so, you know, even though we did set out to replace this, and we have identified some possibilities, it has proven to be very challenging. And so, you know, we're still looking to replace this with something good, something better than the trimethoxy, but we haven't gotten there yet. We thought about trying to alter the hinge binding motif. The hinge binding is through this pyridyl nitrogen in this molecule, and we thought that if we could strengthen that interaction, that maybe we could improve potency. And then the solvent side binding group over here, we've seen that it can impact kinase selectivity and certainly the ADME and solubility properties. When we have this piperazine, we have basic functionality, which makes the molecule quite soluble and also, you know, amenable to salt formation later on down the road. And you'll see that, you know, we may need to take advantage of that moving forward. So, you know, it's taken a little while to finalize this, and, you know, even this is not final. I think, you know, the flow schemes in projects are an evolving beast all the time. But where we are right now is we have 2 sites making molecules here at OICR and at Charles River in the UK. We're making analogs. Both sites then ship their compounds to RBC, Reaction Biology, who test the compounds, and they're actually doing it for free, which is fantastic. They test against the wild type ALK2, ALK4, and ALK5, so the main target, and against the 2 anti-targets. And then at SGC Oxford, they're testing the compounds against ALK2 and ALK5, and nanobret and luciferase cell-based assays, and that's moving along really quite nicely. At OICR, we're taking the compounds and looking at the initial in vitro panel, looking at mouse and human liver microsome stability, as well as CAPO2 permeability. And when we do get the MDCK-MDR1 assay up and running, we'll look at that too. SGC Oxford is also making a lot of progress recently, generating Xray structures of some of our most promising compounds against ALK2. We've got some ALK6, and, you know, I think we still have yet to see an ALK5 structure, but, you know, I think we're generating some really interesting data that's helping. We have a couple of labs, both SGC Oxford as well as Jerome, who are generating data on the phospho-SMAD and ID1 implications of our molecules, looking at target engagement, making sure that we're getting the expected downstream effects that we should see using an efficient ALK2 inhibitor. And then again, you know, both labs are looking at proliferation in DIPG cell lines. I think this is still an area where, you know, you'll notice that the box is dashed. This, to me, generally means that we're not quite there yet in terms of finalizing that assay for profiling our compounds. I think, you know, we have several cell lines that we're looking at. Some of them are more potent than others, and we're still trying to optimize, I think, and extend that panel. I think Javad's lab is also looking at some additional cell lines, so we're hoping to generate something that's sensitive and responsive, really quite responsive to our ALK inhibitors, and as well as I mentioned, I think right up till now, I think we've generated data between 4 and 7 days. The 7-day data appears to look better, but maybe going out to 11, like the SH data, might be something that we need to do in order to get a full anti-proliferative profile for these compounds. Then we move into the in vivo setting. If everything looks good here, we look at the traditional pharmacokinetics in mice, and then if it looks good there, we move into an initial profile looking at the concentration in brain versus the concentration in plasma at a couple of different time points, 2 and 4 hours is what we've settled on. We've identified a couple of kinases that we tend to hit with at least with our lead molecule. So we haven't tested very many compounds in this panel yet, but what we see generally is that our series is really quite clean against other kinases, and I'll show you some data coming up for that. But we can and we should profile more of our compounds through this once we identify additional promising compounds. Now, the ALK2 mutants, so it is the mutant kinases that we are actually targeting that do drive the disease because they are activating mutations. But what we were able to demonstrate very early on in the project is that these ALK2 mutants are essentially equally responsive as the wild type. So we do spot check important compounds to make sure that we're maintaining that activity against the mutants, but we generally screen only against wild type because those assays are much better validated at RBC. Everything looks good here. We move into more advanced in vitro ADME profiling, looking at plasma protein binding, brain tissue binding, and CYP450 inhibition. Safety pharmacology, looking at hERG and Ames and profiling the compounds through the SERA panel, and then a broader kinome profiling if the compound continues to look good. And we have one compound actually that has advanced through all of this, M4K 2009, that I'm going to speak to this flow scheme moving forward, but we've done a dose escalation study as well as a maximum tolerated dose study, which actually turned into a tolerability study because we didn't actually identify a maximum tolerated dose. And then we will move into an efficacy study. We've actually shipped the first compound to the University of Pennsylvania, and they're going to be looking at it in this FOP R206 knock-in model. And then what you can't see down here, I can't anyways, is we'll also move into the relevant DIPG models once we have data in this FOP model to show that we can impact ALK2-driven disease with these compounds. So that's kind of how we're profiling the compounds. So the first thing we did was to start out and to profile a handful, about 30 of the original legacy compounds that were made in this program that led to the identification of 214117. So M4K 1046, it has the aminopyridine, was really quite potent against ALK2, potent in the nanobreath, but really not very selective. And this is something that we've seen over and over. When you add the 2-aminopyridine, essentially you lose the selectivity against ALK5. M4K 1055, in our hands now, this is the 214117. So it turned out to be really not that potent against ALK2. It was 150 nanomolar in the RBC enzymatic assay and 250 nanomolar in the nanobreath ALK2 assay. Now, the compound is really quite selective, but this kind of relatively weak potency might be responsible for the modest efficacy that was seen in the in vivo efficacy study that was carried out. So removing the methyl then and removing the amino leaves you with the unsubstituted pyridine compound M4K 1062, which kind of we felt struck this nice balance between potency and selectivity. And so this is kind of where we started doing the SAR. And then we ended up identifying a very promising compound really quite early on. It was the 9th compound made, and basically what we did was to move the methyl in LDN 214117 from the 2-pyridine position to the 4-pyridyl position. And there you can see that we're really nicely potent against ALK2. That translates into good potency in the ALK2 nanobread assay, and we're 2.5-fold or so more potent than, or more selective against ALK5 than even M4K 1062. So, with a ratio of about 45 in the cell-based assays. Moreover, the compounds were stable in our liver microsome assays. And so we decided to move this compound through our cascade. And so we looked at the pharmacokinetics of the compound. So the KCO2 looked good. It was, A to B of 7.0 and B to A of 9.2. So efflux was really quite low. And when we ran this compound in mice, a full mouse PK experiment looking at 2 mg per kg IV and 10 mg per kg orally, we see that we get a compound that has a really quite low clearance, similar to what was observed with 1055, but we get really good exposure with a Cmax of around 6 micromolar and an AUC of around 52 micromolar, or out of micromolar. And the bioavailability was high, let's just call it 100%. And in fact, this is something that we see with several members of the this class of compounds. So just removing this nitrogen in 2043 gives a compound with a similar overall profile with good exposure. This one actually has a longer half-life with a similar Cmax and AUC and bioavailability. And then substituting the nitrogen here is also well tolerated, and you see that we get a compound again, slightly inferior, but a good overall profile. So we took this data, moved M4K 2009, in fact, all of these compounds into the brain exposure experiment. We see that M4K 2009 gave about a 6 micromolar exposure in the brain after 2 hours, after a single 10 milligram per kilogram dose. And that dropped to about 2 micromolar after 4 hours. And the exposure in the plasma in this experiment was higher, about 2-fold higher than what we had seen in the original pharmacokinetic experiment. So we are seeing some variability in the plasma exposure for this compound. And you'll see moving forward, it's something that we want to kind of get to the bottom of. But we've seen numbers in this experiment that brain to plasma of about 0.4 to 0.5. But in other experiments that we've seen, Charles River, where they dosed IV, we see a 1-to-1 ratio, essentially 100% in the brain. And in fact, the preliminary data that we've generated in our MTD experiment as well also shows essentially 100% brain penetration with this compound. So, the ratios between kind of 50% to 100% getting into the brain, I think, clearly shows that we are getting into the brain with this compound, and the absolute numbers need to be verified. 2043, where we removed this nitrogen, this compound didn't show very good brain penetration. And if I go back to this slide, this is the one compound that did show a little bit of efflux. It's about 2.5-fold, nothing that substantial, but it is present. And so we think that might be the cause of this relatively poor brain penetration, that it is being effluxed. 2045, where you cap this nitrogen, also showed very good brain penetration with ratios between 1 and 0.5. So it's, again, also potentially a very interesting compound, but it is slightly less potent. So we prioritize M4K 2009. We've also been able to show that the M4K compounds do act as they should by decreasing the phospho-SMAD1 as well as the downstream ID1 expression, and they even impact the viability of DIPG cells in vitro. So the lead compound, this LDN214117 shown here, was about 440 nanomolar, looking at the decrease in the phospho-SMAD in the presence of the compound, whereas M4K is about 6-fold more potent. It's about 50 to 70, or sorry, 78 nanomolar. This is the nanobret data. And so the data really aligns quite well with what we're generating in the nanobret data, or in the nanobret assay. And then the ID1 expression is shown here, where relative to DMSO control, we're down to about 30% ID1 expression relative to what we see with DMSO. So we think that that's quite interesting, and this is at 1 micromolar. Now, M4K 2009 does decrease the viability of the DIPG 007 cell line. This is the cell line that has been used in the literature quite extensively for DIPG. There is an animal model based on this cell line, so we feel that hitting this cell line would be important. And what we're seeing is about 1.6 micromolar in this assay, and so this is definitely much weaker than what we would expect based on the data that we're seeing in the nanobret as well as the phospho-SMAD assays. But we are looking at other cell lines. Javad and Jerome are doing that. I talked a couple of times now about looking at a longer time frame, going out to 11 days to match the shRNA. And then it may be that, matching the IC50s may not match this. We may need to actually hit an IC90 to actually have a significant anti-proliferative effect. And so, we're trying to work through this, trying to understand where the discrepancy is and what the difference is between the readouts we're getting in these assays compared to the effect on the proliferation. So this is still a work in progress and something that we're trying to understand.
Sorry, I might have missed it. Was that a 3-day assay or a 5?
This one was, I think, a 4-day assay.
Yeah, I guess this was done by Johnson,
right.
I think, right?
Yeah.
But yeah, in 4-day assays and some cell lines from Stanford, I see almost exactly the same IC50, so it's like 1.6, 1.5 micromolar.
Right. Right. Yeah. All right. So, given that everything continues to look promising, we moved the compound into the advanced in vitro. Plasma protein binding was 91%. Brain tissue binding was high, about 99%. We're not sure about the relevance of that at this point for this particular target. The compound was profiled at Charles River in the CYP, and we didn't see any inhibition of the 5 major isoforms at all, up to about 10 micromolar. We did see hERG inhibition in a patch clamp assay at about, or a binding assay actually, of about 8 1/3 micromolar. So this is something that we'll have to follow up. And the Ames was negative. We moved the compound into a SERAP panel looking at a safety screen 44. We did see a couple of hits specifically in the adrenergic alpha-2A and cannabinoid CB1 receptors, looking at 90 and 97% inhibition at 10 micromolar. We followed this up, and you see that we're around, the IC50s are around 2 micromolar for these compounds. So in these assays. So I think, that lines up with what we see with the ALK5 off-target effect as well. So I think all of that is kind of lining up pretty well. And so we're not sure yet how big of an issue this is going to be, but I think that, we do have at least a comfortable margin between that level of potency and at least the level of potency that it takes to impact the phospho-SMAD. We also looked at the kinome profiling at RBC. We hit 10 of 374 kinases, more than 50% at 1 micromolar. We generated IC50s against those, so the percent inhibition, or the percent activity remaining, is shown in the percent here, and in brackets are the IC50s that we're able to obtain against these kinases. So, ALK2, ALK1, these are ones, and ALK6, these are the ones that we knew we hit anyway, and those are kind of 3 out of the 10. Able, we hit really quite weakly around a micromolar, not really an issue. DDR1, is actually quite interesting, I think, as an off-target effect. I know that Alex has published some papers on this, so we may actually be able to have a nanobret data for this as well, something that we might consider, but we are pretty active against DDR1 at 41 nanomolar. The other ones, again, not that much, and TNIC about, slightly less than 100 nanomolar. So again, I think, hitting these two kinases at less than 100 nanomolar may not be a very big deal. In fact, it might be beneficial. So it's also worth mentioning that we did, the compound was also ran at DiscoverX, and essentially we got very complementary results. In looking at the compound at 1 micromolar, we identified most of these kinases as well. So, I think for a kinase inhibitor, it's quite clean, but still things that we should follow up. And of course, the hERG is probably, I think, the biggest potential liability that we have with the molecule so far. But we don't think at this level that it's a showstopper.
Are you going to follow up with the functional consequences of this case, the energy?
If the compound moves forward, we will. I think at this point, we have this. I don't know if it's worth it at that potency level to continue to follow it up. I mean, we're open to suggestions, though. I mean, if you think we should,
You're right. If you're going to push it forward, just to find out whether they are agonist or antagonist.
Right.
Determine whether or not you have an issue.
Yeah. Yeah. So we moved 2009 into a dose escalation and tolerability studies. The results are shown here. So the compound dose escalates really quite nicely, with the Cmax being almost linear out to 100 mg per kg, and the AUC is almost perfectly linear. It's rare that you see something quite that good, but I think this does speak to the solubility of the compound, at least in the formulation that we're using. And the exposures that we're getting are really quite high, and you can see that as we go higher, the mean resonance time actually gets longer, which kind of corresponds with that longer AUC, even though the Cmax isn't increasing. So pretty promising. We also have looked at the compound over 5 days of dosing, looking at 5 arms. We had a vehicle arm, BID, 25 mg per kg QD and BID, 50 and 100 mg QD. All the doses were tolerated. We didn't see any deaths or significant body weight loss or gross pathology. We collected the plasma at 2 and 4 hours, and we collected the brains at 4 hours. And as I mentioned, the analysis of this is ongoing. Initial observations say that the brain concentrations match the plasma concentrations. In fact, they might even be a little bit higher at the end of the study, which could be a good thing, but we're still working through that data, and we'll hopefully have it for the next meeting. So we didn't see any body weight loss with the compounds. You can see that we didn't gain as much as the vehicle did, although the error bar at this last time point is slightly higher, but we didn't drop into losing body weight. We essentially just didn't match the growth or the increase in body weight that the vehicle gained. So maybe this is related to CB1. It could be, yeah. So, definitely something to keep in mind. Maybe we can have a weight loss drug as well. Okay, so then this is new data showing the rat PK compared to the mouse data that we generated. And again, I think it's pretty comparable. Clearance is a little bit higher. Volume of distribution is definitely higher. Cmax is about 4-fold lower. The Tmax is longer, and the AUC is about 4-fold lower as well. But the bioavailability is still quite good at 96%. So I think generally that the PK in the rat is quite good still. And we were able to dose the compound, as we mentioned at the last meeting, IP. And what you can see is that we're able to get up to 3-4 micromolar with this compound, dosing IP at 30 mg per kg. So, I think this lines up pretty well with what we're seeing with the oral dosing. It might be a little lower, but I think generally, I think this should provide the results that we're looking for. So I think it'd be okay for you to move forward with IP dosing, Jerome. All right. Synthesis of the molecule is shown here. I won't really go through this, but I do want to bring up the fact that we've been able to entice a local CRO to join the effort. And GL Chemtech has kindly offered to make us 50 grams of this compound, for essentially only the cost of goods recovery. So I'll show you that, that I think that's going to really help us drive the project forward in the new year. So the next steps that we have, so we've already shipped compound to the University of Pennsylvania to Dr. Eileen Shore. She has this FOP 206H knock-in model in C57 black 6 mice. This is the same mice that we did the MTD study in. We've talked about a 5-arm study looking at vehicle, 10, 25, 50, and 100 mg per day, once daily dosing for 2 weeks with 8 animals per group. And at the end of the study, we'll collect plasma 2 and 4 hours after the last dose. We'll look at brains 4 hours after the last dose. And a heterotopic ossification will be the endpoint. Basically, they injure the Achilles tendon, look for new bone growth, and over 2 weeks is more than enough time to be able to separate vehicle from compound in this study. We'll look at some histology. It's important to recognize that the formulation is important and how you make that formulation. So Ahmed and we will provide detailed instructions to Eileen in her lab to make sure that we have a soluble formulation that's being dosed to make sure that we're getting consistent exposures. And then Eileen's perfectly willing to have a second follow-up study, and we can plan that based on any results that we see here. We considered having BID dosing arms in this initial study, but Eileen felt that it's actually much easier and the mice tolerate it much better to have only once daily dosing in this model. So we're going to start with that. If we need to go to BID dosing, then we'll see. But as I showed you, the exposure with 100 mg per kg and the MRT in particular are really quite long. So I think QD dosing, certainly at the higher doses, we should see something in this model. And hopefully, we see something even at the lowest dose, and then in the next experiment we can dose down. But I think these are going to be really quite exciting results that we should get early in the new year. I think, as I mentioned, we still want to understand the variability that we're seeing in the absolute exposure. Is this a formulation or solubility effect? So, we're considering evaluating different formulations and certainly looking at different salts, in particular the HCl and mesylate salts of these compounds. These should be really quite soluble in a lot of different formulations, and it should address any formulation solubility issues that we're seeing. As I mentioned, GL Chemtech has volunteered to make the 50 grams, and our plan is to make this compound available to the broader scientific community as soon as we have that. It'll be a potent bioavailable CNS penetrant ALK2/BMP inhibitor, because we do hit some of the other BMP type kinases. Also a DDR1 inhibitor that could be potentially interesting. So, we'll see what kind of data the broader scientific community can generate with this. We still want to better understand the brain PK, and Javad Nazarian at Children's National has this nice model looking at real-time brain concentration. So I think given that this compound is getting into the brain at good concentrations, I think this will be interesting data that we can generate. Javad's lab already has the compound, and I think they're looking at some in vitro experiments first, but we'll see what kind of data comes out of that. And then once we understand that we can impact disease with an ALK2 inhibitor that has the sufficient PK and potency and properties that we need, at least in this FOP model, then we'll feel more comfortable moving it into DIPG efficacy models, because these, at least in my opinion, aren't as well validated. I think that, you know, we need to make sure that the results that we get in any study here, we can correlate back to, you know, efficacy, PK, and pharmacodynamics. And, you know, I think making sure that the molecule is active here first and then moving into here, I think will give us a lot more confidence in interpreting any results that we get in that model. So that's going to move forward as well. And Jerome, you'll see that he has some interesting models and some interesting capabilities that are coming up. And Chris and Engel both have this DIPG, or they have this DIPG-7 DIPG model that, you know, is certainly one that we could move into. So the target product of M4K 2009 is really stacking up to look pretty good and to match what we're looking for in a potential candidate in this project. But until we have the efficacy, you know, we can't nominate this thing yet. So, but it's well on its way, and I won't go through this in any great detail, but, you know, I think that the scale up of M4K 2009 to the 50-gram scale should be ready by the end of February. The FOP study, I think, should be done by then, if not before. And that should allow us to at least make a go, no-go decision, or possibly design another FOP experiment based on the results that we get here. In the meantime, we're going to continue doing, looking at the mouse PK variability results that we're getting, looking at different salts, different formulations. And then if we're a go with this molecule, then the next steps are going to be a, you know, more thorough salt screen, looking at rat PK experiments, a rat dose escalation and rat tolerability study. What would come next? We'd probably take a chance and run the dog PK study at the same time to get the cross-species pharmacokinetics that we're looking for. And then the critical experiment, if we're happy with all of these results, is going to be based on the herd findings, this dog telemetry study. And whether or not we, you know, this is expensive, and we're only going to do this if we are going to move the compound forward. Of course, during the whole time, we'll start to look at DIPG models, and what you can't see across the bottom here is to continue to identify backup compounds to fill the pipeline, to, you know, try to identify something that's more potent, more selective, better PK. You know, I think that this is and without the potential herd liability. So the key areas moving forward with our ongoing SARs to improve the potency and our selectivity, decrease the herd, decrease the potential for the off-target pharmacology that we identified in syrup, although now that we have the full IC50s, it's not that bad. And then the high brain tissue binding is also something that we could potentially address with a backup compound, and we still don't know what the relevance of that will be yet, though. So we have identified, I think, some other interesting compounds recently. 2117, this compound, very structurally related, but we've removed this nitrogen and alkylated this nitrogen, is about twofold more potent than M4K 2009 and more selective, so you get a higher ratio of the ALK5 to ALK2 selectivity, which is quite good. No efflux at all for this compound, has a lower clearance, better half-life than 2009 with a similar volume of distribution, and a similar oral exposure with a slightly lower bioavailability. Now, these compounds that I'm showing here do not address the potential herd liability. In fact, they're slightly more potent. You know, I think if you were to test these more than once, you might get similar results. But when we're moving these compounds forward based on the fact that the herd liability may not actually be an issue, but these do address the improved potency and selectivity that we're looking for. And 4K 2118, also very interesting compound, but similar overall potency against ALK2, but much better selectivity against ALK5. very good ratio. And again, the pharmacokinetics look really quite promising for this compound, very similar to M4K 2009. And then M4K 2143, that combines a couple of the beneficial features that we've identified, at least with respect to potency in the project, incorporating the C4 nitrile rather than the C4 methyl, and then cyclizing this portion of the molecule into this tetrahydromethylene. Gives a compound, again, it's about twofold more potent than M4K 2009. And again, because of that improved potency there, we're more selective, at least in terms of the ratio. And the profile looks pretty good for this molecule. We have submitted this for a full PK experiment. We do have in-house data generated here at OICR that shows that we do get oral exposure with this compound that matches fairly well with what we've seen with 2009. So it'll be interesting to see what kind of PK we get with this compound as well. But all three of these compounds have been submitted for brain penetration studies, so we'll see where we stand with these. We've also moved 5 new compounds into mouse PK experiments. 2143 shown here. Again, it's a nitrile with this naphthyridine, so this is the compound that I was showing you on the previous slide. Also going into the brain. This compound that has a trimethoxyphenyl replacement, 2149, is, not quite as potent and not quite as selective as we'd like, but it does, we think, address the hERG. The compound without the methyl and this functionality did not show any hERG inhibition up to 50 micromolar. So, we think that this is taking a step in the right direction, as well as it's the kind of best replacement that we have so far for the trimethoxyphenyl. So we do want to start moving some of these in vivo and see where they line up. This compound we think is,
You should go to the N method and the isopropyl then.
Yeah,
Those are coming?
some of them are, yeah, we have,
I was debated in wondering if it's enough to see penetration.
yeah, they might, yeah, and we've tried to alkylate that too, but it doesn't, it kills the potency if you alkylate here.
Oh,
And-
okay. You tried to calculate the A1?
Oh, this one.
Yeah.
No, this one is well tolerated, yeah, and I mean, we're going through the various permutations with this,
Okay.
also removing this nitrogen,
Yeah.
like 2117,
Yeah.
and alkylating, right, so there's a couple of things that we're doing around that. 2141, I think, is an interesting compound because it gives it's so structurally different on this side. It's potent and selective. One thing that does concern me is in our hands, the metabolic stability in the presence of mouse liver microsomes is actually quite low. So this will be interesting when we put it in vivo to see if we see this in vitro in vivo correlation. We would expect this to have a very rapid clearance and very low exposure, but I think it's worth it at least to verify that. We have seen projects where that isn't the case, so I think it's always good to check that. 2121, very similar to 2143, except for we have, rather than the nitrile, we have the methyl, so I think this is a nice paired match to be able to look at data. And then this is the nitrile with the carbon and the N-methyl, so the M4K 2117 analog that contains a nitrile at this position. That's again, really quite potent and looks pretty good, at least in the initial. So we selected this one as well to move into mouse PK.
SIP profiling on these fields? Have we done any SIP profiling?
Only on 2009. And then, in all of those initial compounds, we didn't see anything. So this initial set, 2009, 2043, and 2045, these were all ran at Charles River. And we didn't see any SIP inhibition across the series. So, so the ongoing SAR, we're looking at a variety of things to improve potency, to improve the hERG. So we've made some initial observations, and we're making the various combinations and permutations of those with the nitrile at this position, the methyl, as well as the various different things that we found on this side that are tolerated. and as well as the replacements that we found for the trimethoxy. So we're making all of those permutations and profiling the molecules. We're looking at capping this NH to remove the basicity, which should address the hERG. Usually hERG is caused by having a very basic residue a certain distance away from aromatic residues, and this kind of fits that profile perfectly. So removing the basicity should address it, but we need to demonstrate that. We're replacing this whole ring with non-basic substituents as well, and we're looking at that. We're looking at cyclizing, and you saw the tetrahydro naphthalene that look good. We're looking at macrocyclizing in both directions across these two rings to try to, one, improve our freedom to operate, but also, if you can get the angles just right, you can get improvements in potency because of the entropic cost of binding, but also, we might be able to improve selectivity that way as well, or specificity. So, we're looking at options there. We're looking at different replacements at this position. So far, we've generated data showing the methyl and the nitrile are quite good. The chloro is a new result that appears to be quite well tolerated, although not as potent as the methyl, but we might be able to use that in combination with some of the other things that we've identified that improve our potency. So that's ongoing. The ethyl is one that we're still looking at. Cyclopropyl is one that just got submitted, so we'll see. And then again, the microcyclization. Replacing the trimethoxy aryl group is still something that's ongoing. We've identified a couple recently. I think the dimethoxy fluoro is one that, we do lose some potency, but I think we can try to regain that potency by looking at it in the context of the nitrile as well as the tetrahydroquinoline that we have on the other side. I think some interesting results that Sue will likely touch on in her presentation have just come out looking at these two substitutions. So these ones are much less potent, but I think still show some promise, and I think we might be able to improve the potency with that. And then the methoxyamide, we're still looking at ways in terms of can we improve the selectivity and the pharmacokinetics and potentially the brain penetration when we have this type of substitution. The other thing that's ongoing is, we're just starting to think more about is exploring the M4K 3007 scaffold as a potential backup series. This was synthesized at Charles River. It looks really quite potent and selective. It has good PK, but very poor brain penetration. We think that doing SAR around this series could lead to a second series of compounds that have good brain penetration. So with that,
Actually, before you hit that slide, going back, what's the prioritization for this different work here? Like, are you focusing more?
It's all ongoing.
But I know. So is it still like a shotgun approach to all these areas, or are we starting to focus on, improving the selectivity at this stage a little bit more?
I mean, no, I think basically we have enough people working on the project that most of this is ongoing now.
Okay, so
yeah, we have several, a couple of people looking at different substitutions here. We've got Diva, who's working on the kind of macrocyclization here, and David is looking at different substitutions up here. Charles River is looking at different trimethoxy aryls and various different substitutions, and other members of our team, including David as well, are looking at the various permutations and mixing and matching.
So what kind of throughput right now are we getting for all these compounds?
David, do you want to comment on that?
We vary anywhere from about 15 to 25 a month.
That's pretty good.
Yeah. Which is
5 FTs?
Fewer at times. Yes, even.
Yeah, because the 5 includes ADME, PK, project manager, et cetera, right? So, but 5, and then the 2 at Charles River as well, right? So, so basically all of this is ongoing.
By the way, I should know that's from OICR only, plus Charles River contributes another, whatever it varies, right?
Exactly. So the other thing that's worth mentioning is that we do have X-ray structures for some of these compounds, and we're starting to generate some very interesting data. I think we saw at the last meeting that there was a really high quality structure of M4K 2117, which actually is one of the compounds we're moving into the brain experiment. So this is really quite nice. We're getting lots of very good structural information. We just had a meeting the other day with members from ICR in London who are interested in helping out the project by doing some computational chemistry and molecular modeling to try to design new and better inhibitors and to look at the different ALKs and look at the different ways that we could try to build in selectivity. So, I think all of that's very good, and, I think we'll see that, SGC Oxford is generating more crystals constantly. So that's good. I think that's going to help us move forward and design better and more selective inhibitors. All right, so in terms of
before you head on, I'm just going to jump back. Are you guys seeing anything that's inhibiting new compound synthesis? Like, are you running out of ideas yet, or is there anything really, or is it still drinking from a fire hose here?
No, I think there's a lot to do.
There's a lot of space, a lot of.
I would say yes. Yeah, I think we can run for quite a while still, especially on this scaffold. And we are starting to push into new things, which really are kind of underexplored at this point. And I think, as Jeff said, I think there's a lot of this mix and match which needs to be done, because sometimes that, especially in this kinase space, that can have very profound effects, right? Taking the toolkit that you already have and kind of rearranging things within the context can often yield results you wouldn't expect.
Yeah. Well, and the reason I'm bringing it up is, the original grant, look, 2009 is really interesting, you said, but we are halfway through, and at some point we're going to have to start narrowing it down a bit.
Sure.
And I was just trying to figure out where we are, if anything's forcing us to be narrowed up. But around March or April, we're going to have to.
Yeah, I mean, I think the SAR is moving forward as we want. I think some of these new compounds that we're profiling, if they show good penetration, we'll start pushing them through our pipeline.
No, I mean, the SAR actually is giving us so many ideas that I'm a little bit concerned about with just 1 year ago is, I mean, when will we know to focus it in a little bit more?
Not so much. I mean, if you look at what the issue is, I mean, you lead compound actually may crash for whatever reason. Things like herd and stuff, I mean, they've identified features that could potentially be a backup that may actually be a winning compound at the end of the day. So you kind of shut your eye and the lead compound looks good. Things do happen in development.
No, I completely.
And they've identified liability. Now is the time to address them.
Yeah.
When your lead compound crashes, it's always helps when you have something that can just immediately feel.
No, I completely agree, and it's nice that we still have some time to keep on going and exploring different. But at some point, we have a non-scientific restraint, which is basically cash and time, that we have to really, put our chips in on this area and start to focus. Just trying to get a sense for where we are along that line. Sounds like we have nothing's scientifically or chemistry-wise forcing us in any one direction at the time, lots of things to play.
But the effort team focused on addressing issues. They're not affecting analogs just with it.
No, and I get it. I just often something narrows down outside of cash.
Well, there's sort of two aspects. I mean, one is just synthesis of the compounds, and there's plenty of ideas, plenty of compounds coming through. But is there a more efficient way to triage them? I think is the other aspect to it.
Yeah.
And if we're going to get a lot of compounds that are starting to meet our early criteria, that's always something about.
Yeah, we may need to consider, going into brain experiments, maybe cassette dosing or something, right? But, we'll see what kind of data we get out of this other set of compounds and where we are. And then I think we also need to test more compounds against hERG, but they also need to meet the criteria. And so David mentioned that we're doing a little bit more on the, so we have the mix and match effort that's ongoing, but we also have some efforts going on more esoteric type ideas, capping at the nitrogen, and so we're just not sure what to expect there. And so far, the results, we haven't generated a ton of potent compounds. You'll see in Zhang Fu's upcoming presentation. So we will need to adapt. Oh, Diva has a question, yeah?
Are we going to be doing crystallization, determining crystallization structures of our actual hERG channel with our compounds that we're making, so we can see what the potential issue is there, like what's binding to hERG?
I don't think so. I mean, I think the hERG channel is pretty well characterized. Yeah, I'm not sure. I mean, So the brief answer is no. I mean, we could talk about it offline, but I'm not sure that that's going to help in this particular case. Yeah, but good suggestion, though.
If you like product profile, what is the number we're looking for in nanograms? Like, what is the potency?
Well, I mean, the problem is we don't really know, right? Because we don't know how potent we need to be to actually inhibit proliferation of these DIPG cell lines yet, because we just don't have a lot of data. So, I mean, the target product profile says less than 500 nanomolar. I doubt that's enough, right? I think we probably need to be less than 100 nanomolar.
209, whatever, 40.
It's around 50 nanomolar in the nanobread assay, and 79 nanomolar in the phosphosamide assay.
I mean, I guess if you wanted to be conservative, you could say how much above the IC90 and the free fraction.
Yeah.
Right.
Which is a tall order when you're looking at free fraction. But,
exactly. So, and again, we won't know, and I think, this initial efficacy study with the FOP model will help us. I mean, the biology may be different, but the underlying mechanism of inhibiting ALK2, decreasing phosphosamide, and affecting downstream markers should remain the same across the different diseases. So you might need to hit it harder and decrease phosphosamide even more in DIPG or FOP one versus the other, but at least, if we see something in this FOP model, it'll give us a starting point somewhere to start trying to gather that kind of information. Yeah.
Thank you.
Okay, so we've identified M4K 2009, established freedom to operate, and I think, we're identifying a bunch of other really quite promising compounds. And I think, a really key point is that this open science model is actually working very well, and it's attracting a lot of people to the project, and that are willing to help, and it's really been an invaluable resource to have these people join and to help move the project along. I think that's just fantastic, and I think this could be a model for other targets moving forward. And with that, I'd like to thank you everyone for your time and for listening to me, which always turns out to be a little bit longer, but I've had to be shorter the last couple of meetings and go quick. So it was nice to be able to tell the whole story. So I think
see that flag getting more and more crowded with names too.
Yeah.
That's going to be just whatever. Never mind.
Yeah.
I was going to come up with something.
Yeah, no, it is.
At some point, it looks like graffiti on the wall. It's really great.
Yeah, no, things I think are moving along nicely. Okay, so I think with that, we can move on to SGC. Unless there's any questions online.
We lost Alex?
Hello?
Hi, Jeff. Sorry, we were on mute.
Oh, okay.
Okay, hi.
Hi, John.
I'll just briefly go through some of the results of the past 3 weeks or so. So for the past, the latest batch of compounds from both OICR and Charles River's lab, only 2 of them were below 100 nanomolar, showing out to IC50 in the nanobread assay. And on the left, it is the substitution of the methyl group on the hinge binder with a chloral group. Mm-hmm. And the IC50, I would say, is still somewhat comparable to the 2009. And I'll have to check the ALK5, the degree of target activity. On the right is another recent compound, 2171, with a nitrile group, but the nitrogen on the solvent side binder has been alkylated, and the potency is still also conserved. So it will be interesting to check how the off-target activities are. And yeah, the other variants on the trimethoxy group and solvent size modification, they weren't really potent though. Yeah, we can. Next slide. And so I'm summarizing several of the compounds that have been where the methoxy group had been substituted, but they resulted in lower potency. So previously with the amide group, we were able to maintain around like 56 nanomolar potency towards out2, but subsequently the alkylation in 2175, for example, it compromised the activity towards out2, as well as other modifications, for example, addition of the nitrogen, and also the solvent site binding group is also is important, like in 2174 for the out2 activity. So yeah, can move to the next slide. And yeah,
I just wondered, when we were trying all these trimethoxy substitutions, junk food wasn't aware they'd ever tested any of these type of compounds. Were there any plans to make some of these compounds or to make hybrids with the M4K series?
Hi Alex, this is Sue here. So the two compounds in the left-hand box, they are M4K 3003 and 3007. So were made in the early part of the by the guys here. I looked up the data from the nanobret assay. They were both around 30 nanomolar, 36 and 28 nanomolar. So we have looked at, so this was some of the sorts of things that we were doing. Obviously, we don't want to put the X is amino, because what we found previously is that tends to lose the selectivity.
Yeah.
But we have looked at some of the sort of similar sorts of things with having sort of non-aromatic rings there, but most of our attempts to put non-aromatic rings have actually not worked out. So we've not quite had the exact hybrids, but quite closely related.
Yeah. Is it worth making the Novartis compound or the Korean compound on the right without the amino group?
I don't think we've made those.
No, we haven't. No. And it might be interesting to try. Yeah.
I mean, we can have a look back if you like.
2009 scaffold.
Yeah.
Anyway.
We can certainly have a look at those.
Yeah, no, I mean, the Korean ones on the right, we were aware, but, I'm not sure that they address much of, I mean, they still don't have very good ALK5 selectivity. I guess if you get rid of the amino on the amino purity, you're going to lose a lot of potency. But, I mean, it might be worthwhile. I think, we need to get rid of the phenol if we want to maintain brain penetrate. So, I think there's probably a couple of things that we need to change in those molecules to make them amenable to brain penetration. But yeah, I think they're interesting ideas. Yeah.
Also thinking of a crystallographic point of view, seeing how these compounds accommodate in the back pocket. I know we didn't get crystals with the 3003, but obviously if we had a few more variants, we might still try those for crystallization as well. It wasn't really to stimulate, it was only stimulating, just revisiting and thinking what might be useful and what's not.
Yeah, no, I think it's worth thinking about, that's for sure, yeah.
Okay, next slide.
I mean, just to add to that, Alex, we have a tendency, we're looking all the time at some of the other, there was also some ALK1 compounds that I put on an early slide, and we're looking at how we might be able to manipulate some of these structures to incorporate features that we know are active from our current series, pyridine series, that we can then take inspiration from some of these others. And some of them we have been looking at. They've not been all made yet. Some of them might be in the most recent screening, but I'll probably not include those on my slides, but because they're no data yet. But, yeah.
Fabulous. Great.
Thank you.
Next slide.
Yeah. So in this slide, I look at the in vitro biochemical activity of the compounds, like towards out1 and out2, because out1, according to the in vitro biochemical data, the M4K series, like a lot of the compounds, they are also almost equally potent towards out1, and it is important in like the function of endothelial cells. So, but interestingly, when I perform the target engagement experiment in a cell, in a cellular context, I do see that for several of the compounds that I've tested, the activity of towards out2 is at least 2 or 3 times more, in spite of having almost similar out1 in vitro biochemical activities. But I'll be verifying this in a more physiological context in the luciferase assay in the endothelial cells themselves later on. Yeah.
Okay.
Like.
Yeah.
Next slide. Roz.
Hello.
Hi.
Hi. So just for the crystallography for the past month or so, I've put up about 50 odd plates with ALT2. Some of those were for new course screens for some of the 21 series compounds, and then also some fine screens to follow up hits from previous course screens with the compounds on the right. So I've just got a couple of slides with images, and if you go to the next slide.
So 3003 is actually in there. You see that?
It is, but when you see the crystals, you'll, yeah.
Okay. Good news, bad news sort of thing.
Yeah, so essentially we're not, a lot of the hits are very tiny needles. And obviously when you follow up very tiny needles, you normally just get a lot more tiny or maybe slightly bigger needles. But I will be, following up any hits that I get with whatever possible conditions I can. So there are a couple on here, like 2145 that I can probably mount. 2149 as well, I can probably mount, and we have a synchrotron trip on Saturday, so I'll send them down. Some of those needly things could possibly go on a loop, and we'll see how they look. I don't have a whole lot of hope, but we'll see. And then I've got on the next page some fine screen follow-ups. So I just found at the beginning of this week 2158 that it's got a very pretty looking crystal there that I've verified is protein with UV imaging, and it looks like it's really those 3 drops actually are all from the same plate. I'm not sure if how easily the 2 on the right will be able to be mounted, but I'll try. I've got a feeling we might have anisotropy in the resolution because that's a very flat crystal. It's large and rectangular, but very flat, so it might be too short in the one direction, but we'll see.
The 2149 looks
and
promising to optimize.
Yes, 2149 looks like it could actually be. I'll try and mount some of those and see what happens, but that could be worth optimizing or using for seeds as well.
The top one.
Yeah.
Yeah. Good.
So that's kind of, we don't have any, no new actual data this month, but we're constantly putting up new screens. So.
Great.
Jeff, did you have the latest version I sent you, or?
Did you send another version since the meeting started?
No, I sent you one just for the meeting.
No, I don't. Let me, yeah. I can pull it up though.
Okay,
Yeah.
so don't worry. So we had one other slide from Ellie where she was crystallizing ALK6 with a few compounds, and she also had a couple of hits. Remember we had a BMPR1B structure with 2009, I think, originally. So there might be another couple of hits. So those were complexes with FKBP12. I just quickly throw in this slide as an advertisement. We're having a meeting on BMP signaling in cancer, which will cover aspects of DIPG. So we'll have someone from Chris Jones's group come and give a talk. 1st and 3rd of April. Maybe we want to organise an M4K meeting around here, whether we want to do that in Oxford or maybe at the Charles River site. If you guys want to come over again from Toronto.
Yeah, this is something we're thinking of doing, yeah.
Yeah.
We can start formalizing it. April's sneaking up on me faster than I thought.
Yeah.
Yeah.
Actually, we'll have someone give a talk on the M4K, so we can have a think who that should be.
Okay. Yeah.
Okay. That's all from Oxford.
Great. Okay, so next is Sue. Thanks, guys.
Hi. So first of all, I was just going to, I don't think Julia was able to join us, who was looking at the DIPG cell lines with and the MTA with Angle. When I caught up with her last week, she said that they're progressing with the MTA. They haven't got it signed yet, but hopefully we will get something signed soon.
Okay.
So again, just, okay?
Yeah.
So just a general update. There's another 19 compounds being prepared and sent for screening this month. These are generally hybrid, further analogues in the pyridine series where we've replaced the trimethoxyphenol. We continue to try and make some progress there, and as well as looking at some hybrids of the blueprint compound with the pyridine series. So we've now sort of moved our focus to looking to try and identify if we can find a second series, really. I think that's probably where we can start to put some more effort in. One of the things is looking at the blueprint compound, but as I mentioned when Alex was talking, that we do also look at all the other sort of patents and various other papers and patents that have, where there's information on ALK2 inhibitors, and we sort of try and merge ideas across series and see if there's areas that we can work on. But one of the things we have been doing this month is looking more at what we can do within or stroke around the blueprint compound, particularly addressing the full brain penetration of that compound. So the next slide, please. So as far as we're concerned, one of the interesting new compounds here was SK3043. So we made, right in the early days, we made a benzimidazole compound, 3010, which is about 150 nanomolar, sort of moderate activity, but not really where we wanted to be. So we made the same compound with the 4-methyl and the 4-cyano, and interestingly, we see quite a big jump in activity for the compound M4K 3043 down to 21 nanomolar. It's interesting when we put a larger group on the benzimidazole ring, we start to get a change in activity. So putting an isopropyl group on there looked to be reasonably positive initially in comparison with M4K 3010, but then when we made the 4-methyl, we got a compound that was completely inactive, so it's 3029. But more recently, we've put a larger group on there, trying to sort of mimic, again, this is trying to look at the blueprint compound, which has that cyclopropyl amide on there. So we thought if we can put a group that will interact in the same sort of, in a possible lipophilic pocket in the same sort of region. Certainly with the methyl, it's better than with isopropyl, but similar to M4K 3018, really, it's in that same sort of ballpark figure. We haven't made the night trials of either of these two compounds yet, but we're also looking to make some compounds where we've got an additional methoxy on the phenol ring of the benzimidazole. Yeah. So there's a few other ideas around that area that we're looking at, and hopefully some more compounds will come through soon.
Yeah, it's amazing the difference that nitrile makes in this series, actually. Yeah, it's quite interesting. I was quite stunned when I saw that result. Yeah, yeah. Good.
So let's move on to the next here. So we've made a whole range of compounds where we've made, so again, these were looking at some sort of aliphatic sort of groups out in that region, in the interacting with the lysine and the waters around that area. Disappointingly, none of these sort of isoindolines or acyl indolines, isoindolinones or phthalamides, all of those, unfortunately, none of those showed any sort of significant activity. And these range of piperazines that we've made, which was again sort of trying to mimic the blueprint compound, none of those showed any reasonable activity. So although there's a few compounds that probably will go into the next assay, we're not planning on making any more new compounds in that area.
Okay.
So next slide, please. So looking at the follow-up on the blueprint compound, so obviously we've seen that M4K 3007 does show poor brain penetration in the PK studies, and we've seen that it has moderate permeability and some efflux, as Jeff was highlighting earlier. So it's possible that the high PK, pKa and molecular weight can be contributors to this poor CNS penetration. It's quite interesting. We've got a number of different pKa calculators. They all show it to have a high pKa, but some of them suggest that the piperazine nitrogen is the more basic, and some suggest that the piperidine nitrogen on the right-hand side is the more basic. So I think we need to look at both of these possibilities. The other possibility would be to actually have get a measurement of the pKa, but that's probably still not going to tell us which is the basic nitrogen. So just looking through we did an analysis of the actual patent, and the vast majority of the compounds have pKa's greater than 9. But we did identify a few compounds. So if you put a CHF2, so within the red box, the compound with a CHF2 on the piperazine there does seem to lower the pKa, replacing the ethoxypiperidine by a piperazine also lowers the pKa. So there's a few examples here that we could consider they are actually described compounds within the blueprint compound. In the green box on the right-hand side, again, same sort of thing, replacing the ethoxypiperidine by piperazine on the right-hand side, but changing the piperazine on the left, that goes up north, to a piperidine, taking that nitrogen out should reduce, well, clearly removes that potential basic center. So there's a few compounds there that we thought we would look at, and we've just been building up some stocks of the intermediate A on the right-hand side, which I think I mentioned at the last meeting. Is some of the chemistry is quite trivial, and then you get to the final steps, and actually the yields sort of drop quite considerably when you do the cyclization and that. But one of the chemists has been making a reasonable sized batch of that, so that we can start to look at some of these analogues here.
So when you say that this is outside the blueprint patent, is it
That's
outside
correct.
the scope, or is it just not exemplified?
I'm sorry?
Is it outside the scope, the general scope of the patent, or is it just not exemplified?
It's outside of the scope. So they have a piperazine. They only have a piperazine in that position. There.
That'll be interesting then. Yeah, that's for sure.
Yeah. So I think that's definitely worth preparing a compound of that sort.
Yeah. Absolutely. Yeah.
Okay. So if you move on to the next slide. So as I've already mentioned, the making the intermediate A, which is back on this slide again, is actually quite a challenge. So we've been looking at some alternative scaffolds. We haven't actually started any synthesis in this area. We've been sourcing intermediates, looking at compounds that might have intermediates that might have improved synthetic tractability. So we're just in the process of seeing how sensible some of these are. And again, all of these fall outside of the blueprint patent, including the ones, the bottom right-hand ones, where you put a substitution on that ring, and then we know how the effect of having a substituent on the pyridine ring, how that influenced the activity in the pyridine series. So you could, this may well influence the activity here as well.
Yeah, some interesting compounds there. Yeah.
Yep.
Interesting ideas. Yeah.
As I mentioned, we're also looking at other scaffold types and combining in, but I haven't put anything on in these slides this month, but maybe next month I'll put a few more of the ideas that we've been coming up with. And I think it's the final slide. This is a new patent that came out, I think it was last week, or it might have been the week before. So this is a patent from University of Houston, generic structure on the left-hand side. I think this is, I can't remember their name.
Alexi.
Alexi, that's the one. That's the guy, yeah. These guys. So it's a RIPK2 patent, predominantly is what it looks like when you read through it, because they've got compounds that are selective for RIPK2 over ALK2, but there don't seem to be any compounds that are selective for ALK2 over RIPK2. But it's interesting structure. So Sylvia has docked the compound into the 5-0xG, and you can see on the right-hand side in the black at the top, you can see that you get a water-mediated interaction of the carbonyl, and then you could envisage some further modifications that might improve the ALK2 activity, such as putting perhaps a pyridine nitrogen in that pendant phenyl ring, which is below the lysine. Yeah. So
yeah, I wonder, we might be able to get some of these compounds from Alexi if you know, we can have them tested.
I think it would be really nice to be able to test one of these compounds, because it was quite interesting when we look back. Some of the ideas we had been having had quite similar structures to this. The pyridone at the top there was pointing in the opposite direction. We didn't have the aminopyrimidine, but we had some quite similar structures.
Yes.
But,
okay, if you make a list, we can request from Alexi
a
handful of compounds. Maybe we can get them shipped, get a couple of megs, get them shipped here for in vitro ADME to RBC for potency and to SGC Oxford for crystallization.
Yeah, I think it'd be really interesting to get a crystal. Sorry?
Sorry?
I was just checking if Alexi was on the call.
Yeah, I don't know. Alexi, are you here this week, or this time?
No.
I guess not. The one meeting you missed.
We're collaborating a little with Alexi on this series, briefly. So he did send us a couple of compounds. I have to check if we've got UH15-4. I think we had UH15-1. So whether this is a later compound, I have to check.
Just check the patent. We've got a copy of the patent here. So that one is less active. So it's similar. It's similar activity at RIPK2 and
-1 is not active in ALK.
Okay. Yeah, inactive at ALK2. So that's not the best one to choose.
Okay, I think just generate a list of maybe 5 or 6 compounds or something we're interested in.
It's quite interesting actually looking at the compounds that have ALK2 have RIPK2 activity, have a para substitution, or that lose ALK2 activity, have a para substitution on that phenyl ring, which is presumably knocking into that water on the if you go to the top right. And go to the left of that. That's it, that water there. So it's possibly clashing into that water there.
I think the RIPK2 had a alpha C out conformation.
Right. Okay.
So the RIPK2 has an alpha C out conformation.
Yeah. So it's a bit more.
Which allows for substitution at that position.
Yeah. So that's where you can get RIPK2 selectivity versus ALK2, but from their description, there's nothing that shows significant selectivity over RIPK2. But well, you know, if you start looking at around the compound, so you might find something.
Okay, yeah, no, interesting. Definitely worth a shot. Right? Yeah.
Okay, I think that's all from me.
Thanks, Sue. And I think we just have Joanne left.
Yeah, I just have a few slides on some progress on generating some more in vivo models where we can test some of the compounds. So one good tool to have would be xenograftable cell lines. So we've heard about the DIPG 7 cell line from Spain. It seems like we now have another one, the DIPG 36 from Stanford, which has a glycine 328 to glutamic acid mutation. And so what I did here is I injected 200,000 cells in the brainstem of Poxenol D2 mice. This is a xenograft protocol that's been optimized by Michel Manger's group in Stanford and used by others as well. And I tested it on 2 cohorts of mice with 2 different passages of the cells, and both of them really kind of correlated nicely. It seems pretty aggressive, actually. If you think it's injected at Poxenol D2, and all the mice are basically dead by Poxenol D40. So, of course, we could titrate that in various ways, but I think this could be actually good enough. We might think of like starting treatment at Poxenol D14, 10, and definitely at this stage, intraperitoneal administration is feasible. I have also an MTA with the DIPG, with the Angel's group in Spain. I don't have the cell lines in my hands. There seems to be a bit of a delay on that side, but although they might end up testing it over there, right? Yeah. So, which is fun too. And I also have brains from many of those mice, and we're doing some histology to see exactly what's happening. The DIPG 36 was derived from a midbrain sort of, I don't want to say metastasis, but dissemination of the DIPG tumor. And when I dissected the brain, it seems to have spread throughout most of the brain. Doesn't seem to be restricted to the brainstem, but we'll see. So for this, all we need to do at this point is establish dosing, schedule, timing, and then I can define a protocol to get a technical approval from the Animal Care Committee. And so of course I need breeders and pups, and also I think it would fit nicely with the timeline that Jeff showed earlier, that we could do that sometime over the winter. This part of the experiment, yeah.
That's great. We should get this part going.
Yeah.
We can do that relatively quickly. I think we should probably. I mean, the only thing I have to show basically is that it's well tolerated in mice, which we know, although it wasn't done in pups, but so. That should be for this type of stuff. It should be okay. Yeah. And then another model that we might want to explore, although this one might be a little less straightforward, is this conditional knock-in mouse model that I generated, which has the glycine 328 to valine mutation that we can activate in any cell type. And we have something in the publication pipeline now. It's not a DIPG model, the mutation by itself, but there are clear effects on gene expression, some very interesting things in the brainstem at Poxenol D7. So that, this is really something that occurs early in development, probably during embryonic development. So if we want to look, for example, at the ability of the M4K compound to prevent or reverse that, it could be challenging. So that's what I'm saying. It's a complicated model. And then, I'm doing a lot of things to combine with other mutations to get the brain tumor models. But it's something we might want to explore down the line. So I presented a lot of this to some people on the line. I've seen all of these data, and Jeff has seen the paper as well. So conceptually, it would be something similar to what Elise will do with the FOP model, right? Where we can just treat with the drug. Even if it's not a DIPG model, then we can still see whether the drug works. But the xenograft model is definitely kind of like more straightforward, I would say. You can just look at survival as your readout, and then that's it. And that's it for me for now. So, we're going to open the protocol. And also, I would say some of the compounds that you mentioned earlier that you
you're looking at brain penetration, the 17, 18, and 43,
Yeah.
you know, I can set up to test it on cell lines.
Okay, that'd be great, yeah, yeah.
And I also have to think of doing the longer drug exposure in vitro.
Yeah.
I think I can only go to 7 days with the type of assay I'm using.
Okay, yeah, I mean, as long as we see that it improves, you know, in that time frame, then I think that'd be better, right?
Mhm.
You know, and then we can design better experiments beyond that. Okay, that's great. So I do believe that's it for our agenda today, other than, you know, we can go back. There's just discussion now, so does anyone on the line or in the room have anything else you want to add to the discussion or discuss?
Well, I just want to do one heads up for one thing we have in mind for the first quarter of next year is to have a clinicians meeting to bring them together, et cetera. That'll probably be something in around March, April period, et cetera. So that's just kind of a heads up for people to think about, et cetera. And we'll have that. I think that'll be part of the just fits in the timeline of as we start to narrow down what compounds we're going to be moving forward to really get some input there as well.
Yeah, it'd be great if we had the FOP data for that as well, maybe.
Yeah, we did,
Yeah.
Maybe the 36 model as well.
Start to characterize to them what we think these compounds, how they're going to work, and which ones they think are the most useful for their treatment of the patients.
Yeah.
So pull that into the mix.
And we'll be able to offer them M4K 2009 as much as they want.
Exactly. One other thing we have coming up, hopefully in second quarter of next year. Right now with the database, we're probably going to start moving that out to on maybe a monthly basis or something so people can have a static database to pull down. We're trying to, from the M4K part of our open science model, and what we're trying to do is do something more interactive. We have struck up conversations with both the Wellcome Trust and today, hopefully the Pew Trust about model databases that they're doing to make it accessible to people. So that will be something coming down the pipe from a different view. But if anyone on the line, et cetera, has any ideas or input, you know, happy to chat, speak up, and we'll go from there. So moving from this first great model, which will be more static, to something that's hopefully, you know, accessible long after we put down our pens on this project as well, in case we move on to something else that'll still be out there and supported by some other party.
Great.
Okay. Could you for some of you in Vitro studies with DIPG? We will be saying a lot of it is PDG, which is, have you planned any with the clinician strategy coming up, looking at some preclinical combinations of PDGF plus just a sort of...
Yeah, I mean, this is definitely something that, I mean, you know, Chris Jones has looked at some combinations, right? And with the things that targeted PI3 kinase signal. So that's definitely stuff we can look at too. But I haven't looked at that yet.
Particularly some CNS penetrant PDGF, I think.
Yeah.
It has a PDGF component in the clinic, is that?
There is one that is on clinical trial for the DIPG at St. Jude's right now. I haven't seen any results, but I know they have the clinical trial going.
Okay.
I don't remember the name of the compound on top of my head, but it's a PDGFR inhibitor.
Oh,
That's St. Jude. Okay, interesting.
You know how they're dosing it? Is it directly or they're doing?
No, I don't remember, but it's on. You can look it up on the clinical trials.
Okay, yeah, that's important. So doing combination with those clinical compounds and to kind of formulate a strategy for treatment.
I know Alex has looked very briefly at kind of EZH2, right? Not a ton yet, but we haven't seen much in terms of.
Yeah, I know Benostat also was tested. It's the clinical trial, like that's the HTAC inhibitor. With the genetic. I saw some data, and then I think the big problem was the side effects. The toxicity was absolutely terrible. To the extent that I think they stopped the trial, if I understood correctly. Very toxic. Which was, I guess, people expected. So, you know, we could use something like that for a proof of concept, but I don't think it's going to be translatable.
For the EZH2 strategy, I mean, the more you look at that, it looks like you got that mutation in the H3K27. But it looks like you want something that actually, instead of an easiest to measure mark thing, put down, you actually want something that removes the mark. Yeah. Because when I started looking when think of EZH, you look like some of the other stories, some of the EED inhibitors might be a better strategy.
EED, yeah.
Yeah, because they're more CNS penetrant, as opposed to the EZH2 inhibitors. A lot of them have that pyridone, which gives them bad CNS penetration property. But the EED angle to inhibit the PL-CETO complex, those compounds have better CNS properties. So if you want to inhibit EZH2 in the CNS, I think the better strategy would go after EED, because EZH2 requires EED for its catalytic activity. So give that some time.
Yeah, and there are some decent.
Yeah, the EED compounds are nice.
Yeah.
Is anything going to trials on EED, or they're just tools?
Yeah,
right now?
there's a clinical trial. Novartis have a compound in clinical trials.
Yeah, that's a good
Is
idea.
the brain penetrant?
Yeah, the compounds are very lipophilic. They're going to penetrate the brain. They're not looking at brain penetrant disease, but it should be.
Yeah, they haven't shown any data, but
Yeah.
we could test
They're
it.
looking at M4K, which is big spots.
Molecular rate's pretty high though, right?
No, it's for them small. Yeah, I actually have to show it to you. Even if it's
Okay. Okay, good.
One other thing, we have to look at the January meeting, the
Right.
next month. So a couple of wild cards, I know it's normally the first week of the month. A lot of people are gonna be on vacation, there's holidays, et cetera, so.
I think it's scheduled for the second
Is
week.
it second week
But
right now?
we're still considering potentially canceling that one, depending on how if there's any new results that come in this month.
Yeah, just because of the holidays, et cetera.
Yeah.
So we'll give everyone a heads up as quickly as possible. et cetera, but,
We may just have a short meeting if there's interesting results to discuss, but,
Yeah, I mean, especially if there's something on the FOC model,
yeah,
that'd be great.
yeah. But, if not, we'll just wait until February. Yeah.
All right, well, thanks.
Okay.
Jeff, thanks for, again, for leading charge on these meetings.
Great. Okay, guys.
Thank you.
Thanks, everyone.
Thank you.